Good morning, everybody. Welcome to the April RCN webinar. Were you able to see my screen when I shared it a minute ago? Yes? Okay. I'll share it again. I don't know why it said it was paused. I'm having some trouble here. Kevin, would you mind sharing the screen? Thank you. We're waiting for everybody to trickle in here before we get started. Okay, I'm going to go ahead and start and just go through our introductory slides today. So welcome to the RCN webinar. This is a webinar on wastewater surveillance for SARS-CoV-2 and other emerging health threats. Next slide. We're really excited to have two exciting talks today. One is out of the Houston area, Jingjing, who's a postdoc, and Kaavya Domakonda from Houston Health Department are going to be talking about their work with measles surveillance in wastewater. And then our second talk is Marina Mozgovoj, who is from Argentina. And oh, Kevin has the incorrect affiliation. Sorry about that. She is a researcher at the National Institute of Agricultural Technology in Argentina. So we're really excited to have her here talking about the work on enteric viruses. Next slide. I want to remind you all of the ongoing activities here, and Kevin can put the links to those in the chat. And I think that's our last introductory slide. Oh, yes, I wanted to make a pitch for May. We're going to have two speakers. Marissa Eisenberg from University of Michigan will be speaking, and Marlene Wolf from Emory will be talking about HIV in wastewater solids. Okay, so with that, I'm going to pass it over to the Houston group, and they can begin sharing their screen here in a second.
Can you see my screen?
Yes.
Okay, great. So, good morning. I'm Jingjing Wu, and I'm glad to have this opportunity to share our current work on wastewater surveillance for measles. In this presentation, I will cover our assay development, application, and our action plans. I will start with some background information about measles. The measles virus is a single-stranded enveloped RNA. It can be transmitted in the population through direct contact or airborne spread. Measles is one of the most contagious of all infectious diseases, but it's also vaccine preventable. Measles, along with mumps and rubella, can be prevented with the MMR vaccine. The FDA has approved two vaccines for use in people aged 12 months and older. However, the herd immunity, which is also known as population immunity, can only be achieved when more than 95% of a population is vaccinated. According to the CDC report, the national coverage for MMR vaccine was only about 93% for the past school year in the US. That's the third straight year and the rate falls below the 95% threshold. This map shows the MMR vaccine coverage for kindergarteners in each state in the US during the last school year. And we can see here that the vaccine coverage rate was below 95% in many states. And in some of them, it's even below 90%. So because of this, our lab started to pilot the wastewater surveillance for measles last year. We hope that the wastewater data can be used to encourage MMR vaccination in the population, especially when a positive wastewater sample is observed. Another motivation that makes us want to incorporate measles monitoring into the current wastewater surveillance system in Houston is the measles 2024 outbreak. This bar plot here shows the number of measles cases reported by week in the US. We can see an increased number of measles cases and several peaks in 2024. By the end of March, 97 measles cases has been reported in the US, and here is the case map. 59% of the patients were unvaccinated, and 52 of them were under 5. It also shows some news titles indicating that the current measles outbreak is not only happening in the US, but also in other countries such as Europe. Now I will get into some details about our work on measles wastewater surveillance. We first designed a multiplex assay for measles, mumps, and rubella in wastewater using RT-ddPCR. For preliminary validation, we ordered the G-block standards and used them to test the design assays. ddPCR results are shown in this figure. Channel 1 is for mumps, channel 2 is for measles, and 3 is for rubella. This figure shows that our assay can amplify all the G-block standards as expected, and all of our MMR assays were designed by Michael Wang and in our group. After the initial assay tests, we conducted a series of tests to validate our assay in wastewater. In this test, we use the ATCC standard list here in the table, and we aim to compare the MMR RNA abundance in wastewater liquid and solid fractions. We calculate the RNA recovery rate across the wastewater analysis process, and we also investigate the MMR RNA persistence in wastewater samples. First, we compare the MMR RNA abundance in wastewater liquid and solid fractions. The process is shown here. The ATCC standards were spiked into raw wastewater from wastewater treatment plants in Houston, and the sample were allocated into 550 milliliter Forkin tubes. So we have 5 replicates for each measurement. After the centrifugation, the supernatant and the pellets were processed separately. The supernatant underwent the electronegative filtration, indicated as H3 filtration here. Then both fractions underwent bead beating, RNA extraction, and then we quantified the RNA concentration in both the liquid and solid fraction. This analysis process here is the regular pipeline we use for wastewater samples in our lab. So once we have determined the method, we can easily add the measles monitoring into our routine wastewater surveillance system. And here we measured higher levels of MMR RNA in the wastewater liquid fraction than in the solid fraction as shown in the box plot on the right. We also calculate the viral RNA recovery rate by dividing the measured viral RNA copies in the liquid or solid fraction in 15 milliliter wastewater by the number of copies spiked into the wastewater. The equation is shown here. The stored solution concentration are based on quantified heat-lysed ATCC standards. As shown in the table, the recovery rate was between 58% and 70% in the liquid fraction and below 5% in the solid fraction for all 3 targets. So in the following experiments, we choose to use the H3 filtration method to process wastewater liquid fraction for MMR viral RNA detection in wastewater. Next, we conducted an experiment to investigate the persistence of MMR RNA in wastewater. The ATCC standards of measles, mumps, and rubella were spiked into 400 milliliter wastewater samples at 2 concentration levels, low level and high level. Low level is about 10 to the 6 copies per liter, and the high level is about 10 to the 7 copies per liter. We also include the raw wastewater samples as an active control. The wastewater samples were placed at room temperature and 4 degrees C, and for each condition, we had triplicates. The persistent test was planned to continue for 28 days. The MMR concentration is planned to measure on 7 days of them, as shown here. Like on the day of measurement, 15 milliliter of wastewater from each bottle was processed with concentration, RNA extraction, and quantification to measure the target viral RNA concentrations in the wastewater. And because this test is ongoing, so we only have the first week results. The right figure presents the viral RNA decay in wastewater. The decay was calculated as log 10 of measured concentration in wastewater on each day of measurement, divided by the original measured concentrations, which is C0. The figure shows that, in general, the MMR viral RNA was stable in wastewater samples over the first week when stored at 4 degrees C. Based on the 1 week results, we can also see over 1 log decay of measles and rubella RNA in wastewater samples at room temperature. And we see similar results observed for both concentration levels. Because measles has wild type and vaccine strains, the second part of our research is to evaluate the measles PCR assays to distinguish the wild type and vaccine strains. The objective of this part of the study is to design ddPCR assays that can detect and distinguish wild type and vaccine measles strains in wastewater and evaluate their performance for amplifying measles wild type and vaccine strains. For the evaluation, we bought 2 measles ATCC standards. One is the Edmonton vaccine strain, and the other is the wild type strain B3. The B3 and the D8 are the 2 dominant circulating genotypes of measles currently. The basic idea of our assay design is that for ddPCR results, better separation between the positive and negative droplets tend to be observed when the ddPCR assay perfectly matches to the target genomes. An increased number of mismatches decreases the amplitude of the positive droplets. And when the number of mismatches reach a certain level, we cannot separate the droplets, or the target genome just cannot amplify. So in our design, for example, for wild type assays, we aim to decrease the mismatches between our assay and the wild type measles genomes and maximize the mistakes between the assay and the vaccine strain genomes. Our vaccine strain design is just the opposite of this. This slide summarizes the number of mismatches between the current measles design assays, between the current measles assays and the different genome types. We designed 3 assays for the wild type strains and 1 for the vaccine strain. They all have the same forward and reverse primers, but we changed the probe designs to distinguish the wild type and vaccine strains. We aligned all these assays to 2 circulating wild genome types and the Edmonton vaccine strain. The number of mismatches are presented in the table. Our wild type assay1, which is used in the previous validation test in wastewater, has no mismatch with the wild types and 1 mismatch with the vaccine strain. So trying to optimize that for the wild type assay2 design, we redesigned the probes to increase the mismatch with the vaccine strains. And for the wild type assay3, We have one mismatch with both the wild type strains and two with the vaccine strain. We made this design to say if the amplification can happen when there is only one mismatch, but cannot happen with two mismatches. If so, we will be able to amplify only the wild type strains. And then we also use the log nucleic acid for the wild type acid3 design to improve its specificity. We also designed a vaccine assay with no more than one mismatch with the vaccine string and 1 to 2 mismatches with the wild type strings. For comparison, the CDC also has its assays. The CDC wild type assay can theoretically amplify all the measles strains listed here, and its vaccine assay has no more than one mismatch with the vaccine strains, but has more than one with the wild type genotypes. So ideally, our wild type assay can selectively amplify the wild type strains or distinguish between the wild type and vaccine strains. And then we can also use our vaccine strain as another source to confirm the result. Now I'm presenting the evaluation results of our measles ddPCR assays. The results were based on the direct quantification of heat lives at TCC standards B3 and the vaccine strain using different assays. The figure shows the ddPCR results of wild type assay 1, 2, 3, and the vaccine assay. The first two columns are the wild type standards. The next two are the vaccine standards, and the last two is a mixture of both of them. Based on the amplitude levels, you can see that our wild type and vaccine strains can distinguish the two ATCC standards. Although similar results were observed between the wild type assay design 1 and 2, the result for this one is presenting the worst case scenario because it has one mismatch with the B3 strain which is used here as a standard, but has no mismatch with D8, which is the other circulating strain. The wild type ATCC3 only amplifies the wild type ATCC standards as we expected, but differences in the amplitude between the positive and negative droplets were less than the other assay. So our next step is to evaluate these assays in wastewater and apply the assays to wastewater surveillance in Houston for measles monitoring. And I will now hand it over to Kaavya for the next part.
Hey, good morning, everyone. I'm Kaavya Domakonda from Houston Health Department. I oversee the wastewater program here in Houston. So as you'll see the slide, currently we have about 38 wastewater plants that we serve, 12 lift stations, 66 manholes, which include 46 schools, 10 shelters, 8 nursing homes, and 2 jails in Houston. So this kind of serves about 2.6 million population in Houston area. And for the MMR pilot, we are using the 46 public schools data downstream wastewater plants to analyze the data. Jingjing, next slide. Okay, as we are moving from normalizing COVID and looking into new pathogens, what we decided in the Houston Health Department is to do a response action framework internally, how we should address actionability in the community if we identify a pathogen. Here, for example, measles. We kind of categorized all our new pathogens into vaccine preventable, non-respiratory, different categories to kind of address actionability in Houston. So here is, I mean, I don't know how y'all can see, like we kind of work with infectious disease doctor advisory group in Houston to identify which categories these would fall within, whether it's vaccine preventable or non-vaccine preventable. Right now, measles would fall within vaccine preventable. If we identify measles in one-off manhole data, we would share that information with our leadership and see how we would move forward with addressing it. And we involve several people within the department, like our EPI group, to contextualize whether it is a real case, how do we validate it, we are going to work with our comms team, how to address the communication, how we disseminate this information within the community, and also work closely with our immunization team to see how we can set up immunization drives to encourage people to get immunization if there is a measles outbreak within Houston area. So here, y'all can see like we have different groups of people. Now, we also categorized into priority areas, whether it's a high priority pathogen or a low priority pathogen. So there were several people involved in making these decisions within Houston Health Department. This is an internal framework for us to work with our experts within the department to address this situation if ever it comes. Next slide, Jingjing. One of the approaches we made that we are working with schools is text alerts. Right now, we are using this for flu, RSV, when we identify positives in school-based manholes. We have parents sign up for this text alert where they can get an information from us saying we have identified a positive flu sample in your wastewater within the schools. And if there is if your child is not vaccinated, please come to this vaccination site, which would be on a certain day at the school location. And we also tell them some education and provide them some education information. If your child is sick, make sure they stay at home and take whatever is needed for them to help with the flu or RSV infection if they have it. Right now, we have about almost over 1,000 parents that have registered in this school alert, and we continue to work with school system to kind of disseminate the information on how they can sign up for these text alerts. Also closely work with HISD schools to, Houston Independent School District, to see how they can address this issue at the school level by if they have to increase sanitation, if they have to monitor if their children coming sick to school. So there's several protocols that HISD has built, and Health Department has a program that works closely with schools. So it has really helped us improve the process within the school system, like what measures they have to implement if we do identify any positive results in wastewater in schools. And also work closely with the caregivers, like the parents. We have a person that goes out and provides education to the parents on how they can help their kids from getting these infections and what measures they have to take. And when vaccination mobile unit goes to the schools, we not only give vaccination for the pathogen that has been identified in the schools, but we also kind of address other vaccinations. If there is a gap within the vaccination for these children, they get those vaccinations out. That's pretty much it, I think. Yeah, please ask if you all have any questions. I can go into more details.
Yeah.
Great. Thank you, Jingjing and Kaavya. So just a reminder for everyone, if you have questions, put them in the Q&A box, and you can also upvote questions you see that maybe you have the same question or would like to hear the answer to. So I think actually Rochelle asked her question about differentiating wild type and vaccine before Jingjing presented some of that information. So maybe in the interest of time, I'll move on to some of the other questions and maybe come back to that. So early on, why did, this is for Jingjing, did you separate the supernatant and pellet? Were you afraid about losing some of the signal in the solids? And do you have a sense of what % of the signal was in the solids?
So according to our results here, like for the recovery calculation, we see like we can recover more than like nearly close to 60% of the viruses in the liquid, and only like range from 1% to 4% in the solids. So we think that the fraction in the solids is like much lower than the liquids, and I think that's largely because of the total solids content in the wastewater is pretty low, and it's like, if I remember correctly, it's typically like 1 gram per liter. So based on that, we choose to use the liquid fraction to detect this targets.
Great, thank you. But along those lines, there was another question from Marlene Wolf. You showed solids here in copies per liter. Did you compare the solids and liquids on a per mass basis? And did you do anything different in the pre-analytical or extraction steps for the two sample types?
I did a quick quick comparison. Consider that the solid content in wastewater is like 0.1% if we estimate that, and then the concentration difference between here is 1 to 2 log. So if we do a mass-based comparison, we would expect the concentration in solid is about 1 to 2 log higher than the liquid. But because we were receiving wastewater influence samples, like untreated wastewater, we didn't like incorporate the solid test in our pipeline. I hope
Great.
that, yeah.
Yeah, no, I think that's good. And I know folks can follow up if they have other questions too. So sort of along this question of differentiating the wild type and vaccine strains, there's a question here from Pengbo Liu about sequencing, asking to share the sequencing information, but what sequence information did you use to those differentiations? And then along those same lines, for Travis Brown, how would you handle the analysis if you have identification of both a wild type and a vaccine strain within a single sample currently?
For the alignments, I think, because I'm not the person who designed the assays, I think Prashant or Michael in our group can better answer this question, but we got this sequence information from, I think we can get it from the gene bank, and then we use benchling to do the alignments. And for if they are like both presented in our sample, if we see our assay results on these two columns, we have a mixture of wild type and vaccine strains in the wastewater sample in one sample. And for the wild type strains, we can see two levels of amplitude here, then we can quantify them. And then the assay can only amplify the wild type. And then also we can use the vaccine assay to confirm our results if you want to. Does that answer the question?
Yes, I think that's good. And like I said, I know folks can follow up either here in the Q&A or after the fact. All right, maybe time for one last question. And then there's also an answer option in the Q&A box. Jingjing, if you're available to answer some of these when we're done. This is from Noko Munakata. Are the differences in decay rates among MMR significant? And if so, do you have any thoughts on why three viruses might decay differently?
Oh, that's a good question. I'm actually not sure why this happens, and because it's only a preliminary results of 1 week, we cannot see like how does the measles decay would behave in wastewater, but that's something we can investigate later to see why this happens. Thank you so much for bringing this out.
Great. And like I said, there's several questions we weren't able to get to. So maybe folks can either follow up or you can try to answer them in the Q&A if you have
Thank
time.
you.
Great. So with that, we'll introduce Marina to talk to us a little bit about her work.
I was silenced. Okay, I'm gonna share the screen. Okay, can you see it?
Yes, looks great.
Oh, sorry. Here. Well, good morning, everyone. First, I would like to thank for inviting us to tell you about our research in wastewater surveillance of enteric viruses in eastern Argentina with high rates of detection and first report of norovirus GI.5 and GI.20. It is really a pleasure to share our results. So, why to study enteric viruses in wastewater? The study of enteric viruses in wastewater has gained a lot of interest during the last decade, especially after the COVID-19 pandemic, as it allows to detect the viral prevalence, the geographic distribution, the molecular characterization of the circulating strains, regardless of the clinical cases, whether symptomatic or asymptomatic. So, infected individuals excrete large quantities of enteric viruses in their feces for long periods. So, the viral particles could be detected in wastewater. In centralized systems, the wastewater is transported through a wastewater treatment plant, and then it could be reused, for example, for irrigation, or it could be discharged in the ocean. In decentralized systems, this community lacks sewerage network, so the wastewater is collected and disposed into septic tanks or pit latrines or cesspools close to the point of generation. These communities frequently lack also access to drinking water from the network, so they have to extract the drinking water from groundwater using pumps. And finally, there are low-resource waste systems which are completely ineffective, and the wastewater, in these cases, impact directly the environmental waters like lakes, lagoons, rivers, also groundwater, streams, contaminating them. So, wastewater surveillance is especially important in low-income communities, where in these communities, viral infections are underestimated, mainly because of the low detection by traditional methods of viral infections, and also difficult access to healthcare. So in summary, wastewater surveillance can provide viral information regarding viral circulation among a community, not only individual reported cases, also disease information for low-income communities with limited or no data at all, and it can provide an early warning of potential outbreaks and the detection of new emerging variants. So the idea from a One Health approach is to support public health system to make decisions like vaccination campaigns and other interventions to prevent potential outbreaks. Human enteric viruses like hepatitis A, hepatitis E, norovirus, and rotavirus are transmitted through the fecal-oral route. A very small amount of these viruses are required to make you sick, and they are excreted for long periods. Here I highlighted these viruses because I'm going to speak about these viruses in our work. Viruses do not replicate in water or in the environment. They persist and remain infected for long periods, and they are also resistant to high and low temperatures and to common inactivation process like chlorination. This is a picture I took from the CDC webpage. I love this picture because it shows how contagious is or can be norovirus. And it means that a few amount of particles are enough to make you sick. How many? A very small amount. The amount of particles that fit on the head of a pin are enough to infect more than 1,000 people. So what about the detection of enteric viruses in wastewater in Argentina? There are only few reports regarding this. They are mainly from central and northern provinces in our country. Authors have reported the detection of HIV, HEV, and rotavirus, both in sewage and in river samples, and the high prevalence of norovirus genogroup 2 only in sewage from Córdoba province. And in Mendoza, also HIV and HEV have been reported. However, there are no reports in Buenos Aires province. So the objective of our work that I'm presenting today was to track the circulation of enteric viruses in wastewater from 2 low-income neighborhoods in La Plata. La Plata is the capital city of Buenos Aires province in Argentina, and to genetically characterize the detected viruses to gain further insights into their strain diversity and potential public health risk. So here is a map of South America. Here you can see our country, Argentina. La Plata is located at the east area of our country. It has 23 neighborhoods, and Alto de San Lorenzo, which is located at the southeast of the district, is one of the most populated, densely populated neighborhoods that comprises 13 low-income neighborhoods with 3,800 families living there. I want to say that at the time of this study, none of the houses were connected to the sewage network. We collected a total of 100 wastewater untreated samples from the intake pipe that serves 2 densely populated neighborhoods there in Altos de San Lorenzo. The samples were collected weekly from July 2020 to August 2021, and the viruses were concentrated according to the precipitation methods that uses polyethylene glycol and sodium chloride. After an incubation step, at 4 degrees centigrade, samples were centrifuged and the pellet was resuspended in a very small volume in order to concentrate the virus. Viral RNA were extracted using a commercial kit, and the viruses were detected by real-time PCR. Samples that tested positive were further characterized by conventional PCR. What we do is to amplify specific regions of each gene of each virus and then sequence by the Sanger method. Then we analyze those sequences and perform phylogenetic analysis. So, regarding the enteric viruses prevalence, it is shown in this chart that 50% of the samples were positive for enteric viruses, from which 36% were positive for one target, while 14% were positive for more than one target. In this table below, as you can see, fewer samples were collected during 2020 in comparison to 2021, and this was due to COVID-19 pandemic restrictions. Nevertheless, during 2020, ATV, norovirus genogroup 2, and rotavirus could be detected, while in 2021, the same viruses were detected, and also norovirus genogroup 1. HIV could not be detected neither in 2020 nor in 2021. Regarding the occurrence of enteric viruses, you can see here in the left y-axis, the total number of samples is shown, and it is represented by these pink bars in each month in which we collected the sample. And in the right y-axis, you can see the percentage of positive samples in each month. So hepatitis E could be detected throughout the whole period except for September in 2020. Norovirus genogroup 1 was only detected in one sample in July 2021. Norovirus genogroup 2 was detected in most of the months except for July, August, and November in 2020, and in August 2021. And rotavirus was detected throughout the whole period except for May in 2021, and it peaked in these colder months in July and August 2021. So, as you can see, we have high rates of detection of enteric viruses during the whole analyzed period. We tried to sequence all the detected samples, but regrettably, that was not what happened. Here in this table, I want to show you that for norovirus genogroup 2, only 3 out of 15 positive samples were amplified and sequenced. And regarding norovirus genogroup 1, the only sample that was possible, that was positive, could be amplified and analyzed. And regarding rotavirus, only 3 out of 27 positive samples could be amplified. I want to say that we only amplified a portion of the gene that codes for protein BP8, which is a part of protein BP4 of rotavirus. And regarding ATV, only 5 samples out of 25 positive samples could be amplified. However, they yielded a full sequence that couldn't be analyzed. According to norovirus genotyping tool and confirmed by phylogenetic analysis, the detected samples were type as genogroup 2, genotype 4, C220, C217, and G1.5. Black squares represent strains detected in this study, while black circles represent Argentine strains that circulate in the recent years. Genogroup 2, 4, and 7 group with other strains detected in our country, while norovirus genogroup 220 and 1.5 have not been reported previously in Argentina. According to rotavirus genotyping tool and also according to our phylogenetic analysis, 2 of the sequence samples were type as P8. This is a characteristic P-type found in humans, and group with other strains, other rotavirus strains detected in humans. And the other sequence was typed as P3, and this strain group with animal rotaviruses like those found in cats, dogs, a horse in Argentina, and with a strain detected in sewage in China. To conclude, our results demonstrate high frequency of detection of enteric viruses except for norovirus genogroup 1, only detected in one sample, and HIV that was not detected. This could be explained by the implementation of HIV vaccines since 2005 that led to a decrease in number of cases, number of infections reported caused by this virus. Also, this is the first detection of hepatitis E in La Plata. And as I said before, we were not able to have the sequence, so we are not sure of what genotype or subtype are the strains that we have detected. However, other authors in our country reported that the HIV that has been detected in sewage, in wild birds, and in pigs, all belong to genotype 3. And if you remember, I showed high detection rates of these viruses in sewage. So this result is really interesting because notification of viral hepatitis in our country is mandatory. However, only 4 cases of HIV were reported in Buenos Aires during the analyzed period, 2020 and 2021. This could be due to asymptomatic cases, limited report, lack of awareness among medical community, or limited diagnostic ability. Anyway, our results demonstrate that HIV circulated in this community, even in the absence of clinical cases. To our knowledge, this is the first report of circulation of these 2 genotypes in Argentina, norovirus genotypes, GI.5 and GI.20. It is important to mention that norovirus is not a notifiable virus, and cases caused by this virus in Argentina are underestimated, probably due to lack of diagnosis. So, by wastewater surveillance, we demonstrate that norovirus circulated in this community, and also we identified 2 new genotypes, 2 new strains. Regarding rotavirus, despite vaccination programs that were implemented in 2015, rotavirus positivity remains high in wastewater. We detected 2 P types, one that is characteristic and was previously found in humans, and this genotype that is found in animal rotavirus strains. So these high rates of detection of rotavirus could be explained by if maybe there is the possibility that we have not only detected human rotaviruses, but also animal rotaviruses, and even rotavirus excreted from vaccinated children, as the vaccine is an attenuated one. To confirm this hypothesis, we should perform whole genome sequence to characterize them deeply. Results from this work were published in Environmental Advances this year, and they reinforce the wastewater surveillance as a tool, a valuable tool for monitoring enteric viruses, also in low-income or low-resources communities. I would like to thank my work team, Franco, Sol, Maria Jose, Viviana, and Camila. Camila was the hands of this work, also the heart of this work, and our collaborators, especially Belén Pizano and Samuel Miño. And I would like to end with the words of Joshua Lederberg, who won the Nobel Prize in 1958 for his research on transfer gene in bacteria, that said that the single biggest threat to man's continued dominance on the planet is the virus. Thank you.
Thank you so much for that excellent presentation. We have a lot of questions for you, so we'll go ahead and get started. So there's a couple questions in the chat asking about which regions of the genome were sequenced for norovirus and how long the amplicon target was for both measuring concentrations of the virus and also for the sequencing?
Okay, the first one. We amplify the junction region between ORF1 and ORF2 that is frequently used to characterize the norovirus. We did not sequence the polymerase region, but this junction just comprises a very little portion of the polymerase. That was the first question. And the second question, I love the second question because reviewers ask a lot about this in the paper. And I believe it was about the concentration of the viruses, okay?
Yeah, the size of the amplicon. Yeah.
Yeah, the amplicon, the size of the amplicon. It was, I believe, it's a nested PCR, the conventional PCR that we used, and I believe it's 340, the size of the amplicon.
So you use nested PCR to generate the amplicon for sequencing?
Yeah.
And then for the quantitative PCR, did you use quantitative PCR?
No, that was the second question. No, we did not quantify it. It was just a qualitative study. But now for other research that we are performing just right now mainly in environmental waters, we have our standard curves in order to quantify the virus detected, in order to calculate the viral load. So no, I do not have the viral loads of the virus detected. However, we have a table that is uploaded in the journal that shows which cities we obtained. And of course, they were really high. High as over 30, most of them, between 30 and 39.
Okay, thank you. So there's a couple questions about exactly what you measured. So the samples that you processed, were they actually raw wastewater or were they treated wastewater that you presented? And then were the samples grab samples or composite samples that you processed?
Yes, good question too. No, they were grab samples. They were not composite samples, and they were raw wastewater samples. So what we did was to perform the protocol of precipitation with polyethylene glycol and sodium chloride is the classic one that could be found in any paper. But after we concentrated and resuspended the pellet in a very small volume, as few as 200 microliters, I believe, then we performed a first extraction using treacle region. And after that extraction, we extracted viral RNAs using the Chiachen kit that uses columns. So we performed 2 purification steps because we worked with raw wastewater.
Okay, great. And then just another clarifying question on that wastewater. You mentioned that it was from La Plata district with 23 neighborhoods, and 13 of those were low-income neighborhoods. So can you describe, and then you mentioned that those low-income neighborhoods weren't connected to the centralized system. So do you like how many people do you think that system is actually serving, and are they mostly higher income individuals then because the lower income neighborhoods weren't connected? Like, can you describe the population that was contributing to the samples a little bit for us?
Oh, yes, I would love to do that, because it's a question that we ask to ourselves too. I'm not sure of the number of people or families that are contributing to this. All I can say is that this low income, very low income, I don't know if you have seen the photos of the neighborhoods. They all discharge the sewage into a, I don't know how to say it. It's a unique, very deficient network pipe that is like homemade, and all the wastewater were discharged into a common septic tank. So I'm not sure of how many people are contributing to this. I also mentioned that by the time we did the study, they were not connected to sewage network. I'm not sure now. I believe that some changes have been made, but I don't have the exact answer. The Altos de San Lorenzo is a neighborhood that comprises 13 low-income neighborhoods, and these samples were taken from 2 of them, okay? But I don't know exact the number of the family.
Okay, interesting. So there's also a question about like potential animal contribution. So if some of the wastewater from these low income neighborhoods were included in the study, is there cohabitation with animals such as pigs, for example, which we know can potentially be carriers of HEV. What are your thoughts on animals potentially contributing?
Yeah, I believe I will answer first for rotavirus. I am sure that there are other animal rotaviruses that we detected in wastewater, and they are not from humans. And I believe that is demonstrated by the P3 genotype, which is characteristic of rotaviruses found in cats and in dogs and in horses. And as I described, this system is very complicated, and it turns really complicated after rainfalls, heavy rainfalls and floods. So everything is mixed. And if there are animals that are with their owners in the neighborhoods, well, probably we are detecting everything. And regarding HEV in pigs, I would love to have the sequence of the detected strains to answer this question. I think that there is a high possibility that these sequences belong to genotype 3. I don't know if there are pigs that, yeah, they could be there in the neighborhoods, and it's a possibility that these viruses that we detected are from a pig resource also, but a pig source, but I can't confirm this.
Okay, thank you. I guess one more question. We have time for one more question, and I'm going to take the question for myself. Were you surprised that so many samples were negative? Because, I mean, 50% were positive, but 50% were negative. Was that surprising to you or not?
Not at all. I think we did not expect to detect a high prevalence as we detected, as you said, 50%, because we were in pandemic. It was 2020 and 2021 with COVID-19 restrictions. So it was really surprising for us to detect viruses. I believe that maybe the expression of or the infection due to other, in this case, enteric viruses, should have been lower, but no, they were circulating at a high rate.
Mm-hmm. Thank you so much, Marina, for this excellent talk. And it's wonderful to see this work coming out of Argentina, and we're looking forward to seeing what's next for you. So keep in touch. And
yes, of course.
Thank you so much for joining the webinar. And also thank you to our Houston group for joining and talking about measles. Have a great day, everyone. See you in May. Bye.
Bye. Thank you.
Bye.